Hello, everyone, and welcome to today's webinar, Quality Control Processes for Accurate and Reliable Microbial Next Generation Sequencing. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by MicroGenDX. To learn more, visit them at microgendx.com. We encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits window at the bottom of your screen to obtain those credits. I'd now like to welcome our speaker, Dr. Jose Alexander, a clinical and technical director of microbiology, biology, and immunology laboratories at the AdventHealth in Orlando. Dr. Alexander, welcome. You may now begin your presentation, sir.
Well, thank you very much. Especially thanks to MicroGenDX for allowing me to do this presentation. Today, we're going to talk about quality control process for accurate, reliable microbial next generation sequencing. And this is important since NGS is becoming a leading technology for diagnostic infection disease, but especially making sure that the technology is reliable and is accurate is part of the whole quality assurance and quality control process that we need to have. Especially before or during the introduction of this new technology. As a disclosure, I'm a speaker and consultant for MicroGenDX. And the topic that we're going to discuss today is why is NGS relevant for modern microbiology, the important quality control consideration for NGS setup, and interpreting NGS plus biomarkers, especially in the area of orthopedic results. And this is one area that is actually complementing the NGS, especially since NGS result by itself could be a little difficult to interpret, the high sensitivity of the assay itself. So the use of biomarker can help to clarify those results that are probably not completely clinically clear about the outcome and the clinical significance of those results. Before to start, it's important to mention that NGS can complement culture, improve, and also can provide an improvement in diagnostic of infection disease. We know that the current culture have some limitation: intracellular bacteria, nutritionally challenging organisms, fastidious organisms, and also anaerobes. Those are bacterial organisms that require special media to grow. They also require special environmental conditions. And some of them will never grow in routine microbiology culture. So technically right now, the microbiology culture is just limited for whatever can grow in the media and it can grow in the conditions that we provide in the laboratory. The complementing traditional culture with NGS can also facilitate a rapid turnaround time compared with regular culture. If we have a sample that we see organism in a gram stain, and normally will take 24 hours or a little more to grow and to be able to be identified. With NGS, we're going to be able to have this rapid identification directly from the sample. Identify organism that is excluded by culture. How we mentioned before, intracellular organisms that are nutritionally challenging, especially those that are not considered during the setup of the culture, can also be identified by NGS. And one of the areas that is probably the most important is with mycobacteria and fungi. We know the AFB can take up to 8 weeks to grow, and fungi culture are normally kept in the incubator up to 4 weeks. Having NGS able to identify mycobacteria and also fungi can facilitate and provide a rapid identification in just a matter of few days, 3, 4, or 5 days, compared with traditional culture. The other advantage is that having a positive smear, in the case of mycobacteria, a fluorescence smear, a Seal-Nielsen, or a specific fungi smear that are positive to direct observation, beside the culture, NGS could also be performed and have a rapid identification of this organism using one of the gold standard in this case, that is the NGS. But the quality control, but the NGS implementation required a good quality control. And this quality control and the whole implementation can be described in a series of steps. One is the implementation itself. We need to have a validation process, and we have to have a database that is reliable. So before to start with implementation, before to start with the validation itself, we need to make sure that the database that we are using for identifying these organisms detected in the sample is reliable, is accurate, and the sequence that are present in the database are being able to be analyzed and be proved that are able to identify the right organism. And then come the validation process. That we know that there is a different type of validation process depending if it's a validation itself or it's a verification. The sample testing. We need to have processes that are in the pre-analytical step, in the analytical, and in the post-analytical step. We have to consider the importance of the quality of the sample. NGS, like any other microbiology method, require a high quality sample, and we need a sample that is representative. Again, NGS is highly sensitive. If we are not careful with the type of sample that we analyze, we can be introducing organism, contaminant, or even normal flora organism that can make the interpretation of the result a little bit challenging. The laboratory process, they should be able to guarantee that there is no inclusion of any contaminant during the analytical process, especially during the setup process of the sample. We have to have a strong bioinformatic analysis that is able to analyze all the results coming from the sequencing and being able to put in a result as a post-analytical step that is clinically significant. If this is only NGS or if it's a result that is done in conjunction with biomarkers. And then we also have to have an ongoing quality assurance process. Proficiency testing is critical. Every laboratory that is CLIA certified need to be enrolled in a proficiency testing program, and this requires that standardized specimen arriving to the laboratory can be run, and it can be proved that the method is reliable and is able to identify the organism present on the specimen. So when we talk about NGS, in this presentation, we're specifically talking about targeted NGS. And this is the NGS that target the 16S ribosomal RNA genes. Those are genes that are in the bacterial chromosome that codify for the ribosomal RNA. And the idea to target this region is that they are highly conserved genes. And they actually also have some 9 hypervariable regions that can allow us to identify different genus, but also species of organisms. One of the advantages to target genes is that allow us to target with specific set of primers this gene, amplify the products of this PCR, this amplification, and that can increase the amount of sequence from microbial DNA when we compare with the host DNA. How we know when we run sequencing directly for patient sample, we have a lot of DNA from the host, and this DNA has become an interference during the process. So targeting an NGS, targeting these regions, allow us to increase the amount of these genes, when we consider that in relationship with the host DNA, it's a little bit easier to perform the sequence and identify the organism. There is 9 hypervariable regions, but the region 1 or 2 are the one that has the best identification for human pathogens. The same happens for fungi. We have the internal transcriber spaces, and they are also part of the ribosomal genes. And those are regions that allow to identification of yeast, but also molds, are able to identify the genus and also the species for this group of organisms. But it's also important to consider during implementation that There could be a lack of resolution of discrimination between species when we use 16S and also when we use IPS for fungi. The best example is when we have E. coli and Shigella. Those two organisms have very similar, genetically speaking, and some of the sequences are unable to differentiate these two species. So the way to differentiate them is to develop a set of primers that is able to be used not only as a quality assurance process, but also to differentiate between species that are close related, where the NGS is having the most technically challenging to differentiate. An example of that is creating a primer for E. coli or a primer for Shigella. When the NGS detects an E. coli or Shigella, the PCR can solve that discrepancy. And that can not only happen with this organism, but also including with mycobacteria and fungi. One of the important things with 16S is that it's not only for gram-positive, gram-negative bacteria. We are also able to identify partial acid fast and also mycobacteria, any other, including AFB tuberculosis, mycobacterium tuberculosis. So before the implementation, it's very important to have clear what is target, the gene that we want to be able to identify. And then based on that, being able to have a database that is accurate and is reliable at this level. So an accurate bacteria and fungi sequence is important at this moment. So there is the reference sequence that we can find the National Center for Biotechnology Informatics, or in CBI. It's an open access, a public library that we can find sequence of nucleotides that can identify different organisms, bacteria or fungi. But it's important that when we take that information from the database, we are able to review and remove those sequences that are incomplete or those that have taxonomical information that are incorrect. This is a public database. It's important to have in consideration the quality of those sequences that are in the public database, since we can have some incorrect or incomplete sequence in this database. We have to remove low quality reads, also in those regions that are important for us to target. If we targeted the V1 and V2 region, we need to make sure that the quality of those reads are high enough and good enough to correctly identify the species that we're looking for. And we also need to eliminate inaccurate matching species. If we have some sequence that is being published for a specific organism, but when we test those sequences and are not able to identify the organism that actually they describe, we need to make sure that those inaccurate sequences are removed. One of the important things, something that we are used to in the laboratory, using ATCC strains, and this is a valuable source for also verify some of those sequences. If we have an ATCC strain for a specific genus or species, we can run in a sequencing and compare that against the sequence that is public in the database. And for the validation process, so we know that we have a validation and also a verification process. So what we understand in the micro laboratory as a test verification is those that are FDA clear and approved methods. We require analytical accuracy, analytical precision, and reportable range. And this is part of the CAP checklist, the COM 4300. But when we are trying to implement an NGS, we are implementing a lab developed test. There is no an FDA clear platform or FDA clear process to be able to implement NGS for clinical purposes. There is work being done in that direction. Is hopefully in the future we're going to be able to have some platform and some process and tests available that will be FDA approved, and that will simplify the process of verification. But in the process of validation, we need to include also, besides the same three characteristics for the verification, we need to include analytical sensitivity, analytical specificity, and also any other performance characteristic to require that we can include there. Interference substances, that probably will be one of the most significant based on the type of samples that we are intended to use for the NGS in the clinical setting. And this is for the CAP checklist, COM 4350, and that can be found in the all common checklist. One of the first steps for sequencing, and something that we need to reinforce and make sure that there is is probably a highly critical information that need to be transmitted to clinicians is the sample collection. NGS, like any other laboratory result, require a high quality sample, and this is critical. We need to make sure that the specimen that is collected is representative of the lesion, of the representative of the area that we're trying to rule out the presence of infection. We need to make sure that there is no contaminant for normal flora or any other during the process of collection. So it's important to consider too, if there is a suspicious typical organism that we're looking for, that we are able to collect sample for the right source. So in this example, we have a joint infection. So we can collect sample as a synovial fluid. We can have scraps from any joint prosthesis. We can also get sample directly from the intracanal from the bone, or we can collect tissue. So all those high quality samples are critical. And if the specimen is not collected with careful and the specimen is contaminated with surrounding microbiome, those organisms can be present on the final result, and they can provide a little difficulty to the clinician to be able to differentiate which of these organisms could be a real pathogen or which of them could be just a normal flora or colonizer. So sample collection, sample quality is highly important, and this is a responsibility for the provider and for the clinician that is requesting the NGS to make sure to provide a high quality sample, and the laboratory can then provide the rest of the high skillful technique to make sure that we don't introduce any contaminant during the analytical process in the laboratory. So we have to ensure a high quality and reliability of NGS. There is multiple controls that are being introduced during the whole process of NGS. During the DNA extraction, we have internal and positive and negative control that are introduced to guarantee that the DNA is extracted from the specimen. We have 3 levels of control during the sequencing. We have a high, a medium, and a low positive control, and we also have a negative control. This guarantees that the different level of concentration of the DNA are able to be analyzed and recognized and also detected, but in the same time, this also guarantees that the instrument is working at the limit of detection that is expected. We have environmental controls that are set up during the process, but also something that in laboratory we know as a swab test. The environment, the equipment are routinely tested to look for some potential contaminant from the environment. And this environment, this contaminant or these organisms present in the environment are recognized. The laboratory keeps track of them, and those are taken into consideration when the test is performed and the result is obtained from the sequence. Because those environmental organisms, some of them are prevalent in the environment, and we need to make sure that we are recognizing them and we know the level that they are normal in the environment. And then we have a bioinformatics pipeline. We have pre-analytical, denoising, and also diversity analysis. So it's important that all the clinical, the digital data coming from the sequence is properly analyzed. The noise coming from this data is clear, and we can use this data then when the quality is acceptable to start assigning organisms based on the sequence that were detected. So the target NGS, we also have the amplification itself is a process for quality assurance. So all the samples that are batched together, they are barcoding. So we have a specific sequence of known DNA that are attached to every sample, every DNA for every sample. That also, when we have an amplification of these target genes that are the 16S, they allow us to remove, or in relative terms, to remove the human DNA by amplifying the microbial target. And also we have a gel electrophoresis for size selection and quality control. We make sure that the size of this sequence that will be analyzed are able to have the quality and the size that is appropriate for the platform that is being used for sequence. And this is important that every laboratory that is implementing NGS recognize what is the requirement for each of those NGS sequencer platform to make sure that the quality and the size of the sequence actually match what they recommended by the manufacturer. So we have in each plate, especially when we set up a plate, it's important that we are able to set 3 levels of positive control: low, medium, high, but also have a negative to be able to evaluate the potential inclusion of contamination during the process. So when the data, all the information comes from the sequencer, when the sample is run in the sequencer, all that clinical, digital data is now going to the bioinformatic pipeline. So we have a pre-analytical, denoising, and diversity analysis. And then bioinformatic quality control, we use the FRED quality score. The assurance that we have a 99.9% call. And this is something around 1 in 10,000 errors in a nucleotide in a sequence. So we're talking about that we are analyzing fragments of DNA, around 300, 400 base pairs. So 1 error in 10,000 base pairs is what is acceptable. So we're talking about 1 error in multiple fragments. So it's a very minimum error that is acceptable during the sequence. And then that actually will pass the quality control of the digital data to be able to get into the bioinformatic analysis. Chimera detections. During the amplification of this 16S gene, there could be some mismatching or some amplification that stopped earlier, some amplification that can actually combine different amplification fragments, and they give some chimera. These chimera are detected during the process of before to be able to assign organism to be able to avoid errors. So the chimera is important to be able to remove those false positive organisms, to able to provide an accurate on the assigning genus and also species to the organism that are detected. Also, sequence that don't meet the size selection criteria, those sequence of fragment of DNA that don't meet the criteria for the sequencer, or they are smaller than what is expected, they can be removed from the analytical step of the sequencer. Also, any sequence that have an error, not only during in the fragment of the gene that was sequenced, but also in the adapter that were attached during the specimen processing, or even on the barcoding, those are also removed. So from the post-analytical and reporting, we make sure that when the all bioinformatic pipeline is analyzing the data, the quality controls are resolved as expected. The negative extraction control is also as expected, and there was no introduction of any contaminant. There is no template control. That means that those blank samples that were introduced in the batch, they are negative. There was no introduction of contamination during the process. This guaranteed that every organism report from a sample actually the organism is actually present in the sample and was not introduced by the laboratory during the process. There has to be a review of each report and looking for any potential contaminant. If there is a trend of some organisms that are present in multiple results, those have to be reviewed. And those sequences that are unique, those sequences that are not present, or those sequences that are not matching any other reference in the bioinformatic pipeline, they need to be investigated. So there is an automated quality control process that happens in the bioinformatic pipeline, but it's also a manual review that is done by an operator, in this case, a molecular technologist, that is able to review each of those results coming from the sequencer before to be released in a report. So this is a very strong and robust process. not only from the moment that the specimen arrives during the analysis and in the bioinformatic pipeline. So the whole process is to guarantee that the specimen that we are receiving for analysis for NGS, the result that we are reporting is actually from organisms present in the specimen. So those organisms were not introduced in any point during the pre-analytical, analytical, or wasn't any error during the bioinformatic pipeline. So the laboratory needs to be able to provide an accurate, reliable result, trusting that the samples that were received is a high quality sample and the organisms present are clinically significant. Also, a part of to keep NGS implemented in the clinical setting, it's important to be enrolled in proficiency testing. There is multiple organizations that provide proficiency testing programs. They are blinded samples. As a laboratorian, you are very familiar with the proficiency testing of PT samples. They are blinded samples. Every laboratory need to be able to test multiple times a year, and also there is some inter-agreement between laboratories. When the result is released by this organization, they compare against each other and see how well you were able to perform. And this is important to have as part of the quality assurance process. This is also to guarantee that we are not introducing contaminants. We are actually able to identify the organisms that are present in the sample, and we are able to report or able to do what we are expecting to do. And make sure that the whole process not only have a high quality standard, but also is clinically significant when we report it. One of the important areas currently for NGS is the use of biomarkers. An example with MicroGene result, that is an NGS plus biomarkers, and this is offered for orthopedic results. And in the 2008 International Consensus Meeting, that was able to introduce the NGS as one of the criteria, especially one of the preoperative diagnostic, one of the interpretative diagnostic. We can see that there is multiple criteria for calling prosthetic joint infection. We have minor criteria that use biomarkers and assign a possible score, and every patient, when we assign the score, we have a possible decision making. So in the section that we have, when we have the preoperative score, we have the positive histology, the culture, and we also have a positive NGS. All of them have a possible score, and that give a patient a score that give you a cumulative patient score that can provide a probability of infection. So in this case, we are giving the clinician not only a positive NGS indicating, yes, there is organism present in the sample, but we also giving the clinician a probability of infection that is based on biomarkers that is an indicative of how the patient immune response or how the patient, the host in this case, is reacting to the condition that is happening on the joint. If this is a real infection or is not. If the organism present came from a good quality sample collected, and this is translated with a call by the NGS, but also with positive biomarkers. Or the organism present on the NGS are significant, but the biomarkers are negative. So the combination of these two characteristics, plus the clinical perspective that the clinician has, can provide a probability of infection. So the future of NGS with biomarkers seems to be a route that can facilitate the implementation and also the interpretation of the NGS result for being to decide the significance of some of the lesions and if those in the probability of infection in those areas. So in summary, next generation sequencing play a complementary role for infection disease diagnostic and also possible for management. There is multiple processes that guarantee accuracy and reliability. A good validation process, a construction of a verified sequence, a good database, the importance of the quality sample collection. This is critical. As a laboratorian, especially as a microbiologist, we know that we need to have a good quality sample. any good result starts from a good quality sample. And we need to follow this in NGS and even make it even more, even stricter than regular cultures. Laboratory process to avoid and detect contamination is part of the quality management system and is also critical to be able to keep the high quality. Bioinformatic analysis, we have to have a pipeline that's automated and is able to identify any deviation of quality control and also any errors. And the post-analytical reporting is always important to have a human factor to be able to review results and trying to identify some variation that the automated system are not able to. The proficiency testing is also critical to make sure that the process continue as we intended to be used, and as part also of a clinical implementation as being a CLIA certified laboratory. And again, the biomarker for interpreting NGS, especially in infection diagnostics, seems to be playing an important role, and this is a section, an area that could continue expanding to not only on prosthetic joint infection, but also from any other source of infection, and can facilitate, especially to clinicians, to trying to adjudicate probability of infection based on NGS and biomarkers. So in this way, I finished my presentation, and now we're going to have a section of questions. If you have any specific question about this process, I will be happy to answer. Thank you very much.
Thank you, Dr. Alexander, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. And it looks like we already have some great questions coming in. Our first question that we got from our audience is, culture methods grow whatever organism is in the sample. Why is there a need for NGS when culture can already identify the infecting organism?
Well, first of all, we know that organisms that grow in culture are those that are technically able to grow in the media and in the environment that we are setting up the culture. So we don't really able to grow everything. And depending on the specimen, depending on the organism that we're looking for, there is some fastidious organisms that probably are not be targeting in some particular areas. And also there could be some intracellular organisms, intracellular bacteria that we're not going to be able to grow in routine micro media. So we can say that one of the advantages of the NGS is they give you this broad range of detection that is go beyond the limitation of the culture. The second one is if we're talking about organisms as fungi or even AFB, we know that those types of culture can take weeks before we can have a positive growth and even identification. So the other advantage of having NGS, especially in AFB and fungi, is being able to reduce the turnaround time of identifying those organisms. And in the case of fungi, especially molds, we know the molds are technically challenging for identification. We need staff that have experience and expertise in identifying microscopically molds. We need to have the environment for them and the special media. So that's one of the advantages to the NGS is a gold standard method, is an agnostic method that will be able to provide identification of any of the mold present on the sample in a short period of time without requiring all the technical expertise that a mycology laboratory can require.
Thank you. Our next question, which CPT codes do you use to bill for NGS-based identification?
That's a great question. One of the limitations that we have right now with CPT codes and NGS is really there is no direct generic CPT code that can be used. Some NGS laboratory providers are working directly with CMS to trying to get a specific CPT code for the platform for the test that they perform. And this is one of the limitations that is right now affecting NGS in the whole implementation, especially we see them from the perspective of an outpatient setting. In the hospital setting, probably it's a little bit different based on the way that hospitals and tests are actually billed. But from a perspective of a direct payment for the test using a CPT code, that is a lot of limitation that the sequencing have right now. Some sequencing, especially 16S, can be used using multiplex PCR panel too. They can be used as part of the quality control process. And this can be used also as part of maybe a faster identification of some of the most common pathogen based on the sample. Those normally have CPT code, but the NGS by itself doesn't have right now.
Thank you so much. And thank you to our audience for these great questions. What would you recommend as the positive controls for the assays you described? Whole inactivated bacterial and fungal organisms in specimen matrix? Any other formats?
Well, ideally, the idea of a quality control is you're trying to be as close as possible to match the specimen that you will be performing. You have a series of positive controls, and some of them are standardized controls, especially those that are known concentration, like we describe about having high, medium, low concentration for positive positive quality control material. If it's possible, use metrics. Let's say that you're running a sequencing from a bronchialveolar lavage, and you are able to use known metrics to develop those quality control, especially for the extraction portion. I think that would be ideal. The other portion is if you can use live organism, or could be inactivating organism, but ideally would be organisms that are they have an entire structure that haven't been compromised. And that way we can evaluate better the extraction protocol. That would be the ideal scenario. If you are processing a specimen looking for bacteria and fungi, ideally a quality control material should be able to include, or you should include some of the bacteria, but also you can include some of the mold, especially during the extraction protocol, because we know that they have different cell walls, they have a different structure, and some of the extraction protocol need to continue being overseen and evaluated during the whole analytical process of the sequencing.
Thank you so much. Our next question from an audience member: molecular methods are incredibly sensitive. How do physicians separate contamination from sample collections or contamination from laboratory processing?
That's a great question. One of the way that we can differentiate that is, one is based on the concentration of the organism, or actually the DNA observed on the sample. That could be a way to differentiate that, but that's a little difficult. One of the things is a laboratory need to be able to provide a reliable result. So laboratory have a series of quality control process that when the result is released, we guarantee that those organisms were not introduced in the laboratory side. Now, the other variable that we have is where they're introduced during the collection. And that's a highly probability too. If the specimen is not a good quality sample or wasn't collected with a good technique, we are risking the possibility to be identifying organisms in the sample that don't come from the site of infection. And this is probably one of the biggest areas that as a microbiologist or laboratorian, especially implementing NGS, we should make emphasis to clinicians and providers that the sample collected and submitted need to be a high quality sample, and the sample need to be collected at the best technique possible. Because in that way, the interpretation and the use of that result for clinical utility or clinical management can be guaranteed. So the way to eliminate possible inclusion of contaminant during the process is having a robust, strong quality control process and surveillance process during the whole analytical, pre-analytical and post-analytical. But to avoid the confusion of introducing contaminant during the collection is something the laboratory should be able to provide education and training, and especially a protocol for collection to providers to minimize the risk of contaminants or inclusion of sometimes just colonizing organisms.
Thank you so much. Now, how do you identify RNA viruses? Is there a need for an NGS for those viruses? Is the host response biomarkers, are they RNA or DNA based?
Um, we are targeting technically ribosomal RNA genes. So in the case of viruses, the NGS using 16S or RNA-based NGS is not going to be able to identify viruses. So the best approach in this scenario, if the idea is to provide a comprehensive identification that go beyond bacteria, mycobacteria, and even AFB, but also start introducing respiratory viruses as many of RNA viruses as many of the respiratory viruses, is probably the introduction of primers and develop a PCR platform in conjunction with the sequencing. 16S as a sequencing based on ribosomal RNA require amplification. This is a targeted NGS. They amplify this ribosomal gene from bacteria and fungi. During the process of amplification, some variation of the PCR can be developed, and they can be introduced in target primers to target RNA viruses. But the sequencing itself, the genes that are targeted are only bacteria and mycobacteria, fungi. So it's not possible to identify RNA viruses using 16S.
Thank you so much, Dr. Alexander. And we have time for a couple more questions. NGS has given me a ton of data. How do I, as a physician, take clinical action?
Well, there is a lot of perspective that have to be put together when the result is being evaluated for clinical utility. The test shouldn't be seen just as a result, and obviously the data and the clinical management or treatment should not be only based on the NGS result. The clinical presentation of the patient is one, so the symptoms of the patient, how those symptoms actually match with the probability of infection, and with a probability of infection, what actually the NGS results show. The other point important is make use of biomarkers or just simple indicators of possible infection. One of the best examples is, for example, if people want to do NGS from urine sample, that can be a little messy with all the organisms, but a good collected sample, a catheterized sample, or even a catheterized sample used in conjunction with urinalysis can provide information beside the clinical presentation. So this will be important variables to take in consideration: the clinical, any presence of biomarkers, and the result itself. Now, when we evaluate the result from the sequencing, we need to make sure that the laboratory is reliable, especially the process is reliable. We are not introducing any contamination during the process, and the laboratory should be able to provide that. A result should not be released if there is any concern about contamination. And the other point is the sample itself. If the provider reviewing the result was the same provider who collect the sample, it's based on the providers on consider if the sample collected was significant, if the sample collected can actually have some concern about possibility of contamination during the collection. So all those variables need to be taken into consideration. It's strongly recommended that only an NGS result is not enough to be able to have a whole clinical management on a patient. Clinical presentation, possibility of biomarkers, and also quality of the samples submitted should be three variables to be used in consideration.
Thank you so much, Dr. Alexander. We have time for one more question. This audience member says, I would like to have NGS available to my clinical staff and laboratory. Is it viable for a hospital-based laboratory?
I mean, could be viable, but one of the strongest, I will say barriers, but also challenge the laboratory has is implementation of a strong database or bioinformatic pipeline. I think that's one of the biggest limitations. We have an amazing platform currently in the market able to provide sequencing. There is protocol described, and even the company who provide the platform, the sequencers, are able to provide papers about different protocols for sequencing, extraction, and sequencing. But one of the biggest limitations is to have a curated and strong database. There is public database, but again, that database needs to be taken very careful because it's an open Technically, everyone can upload sequences as, to this database that can be used as a template for identification. If we use a sequence or we recognize a sequence that is not common in this database, we only have 1, 2, or 3 sequences and they are poor quality, we could be misidentifying organisms. So hospitals, I think that at that point where we get to the final step of the analytical process, that is the bioinformatics, the data analysis, that is probably the biggest barrier that we have. We need to be able to not only have molecular technologists or even microbiologists who work in infection disease, but we should be able to have staff that is dedicated to bioinformatic analysis to be able to provide statistic analysis and provide an accurate identification of those organisms. And I think that that's probably the biggest barrier that we have, but it's possible. The other opportunity is some agreements or alliance with some company that can provide the data analysis based on a cloud-based platform. So the hospital have all the setup working with this company when the data can be submitted to a cloud and the cloud can send back the result because the database is performed, the analysis is performed in the cloud with these companies. They actually have curated and review it and very well kept databases for the analysis that is done in the local hospital.
Dr. Alexander, I want to thank you for your time today. Would you like to provide any closing remarks before we go?
Yes. So one of the important things with NGS is we know that NGS is a field that is growing in infection disease. It's something that we need to learn, just like we did with, we have been doing good culture with many years. And it's also important to consider that there is a lot of things that we need to learn. There is things that we need to improve. There is areas that we need to explore. We need to find significant for organisms that we've been seeing now for first time. That's something that the microlab also observed with the introduction of the MALDI-TOF. We were used to only base our identification on biochemical reagents, and now when we have the MALDI-TOF, we start seeing new organisms showing up, organisms that we never heard before. So the same situation will happen with NGS. We should be open-minded, but in the same time, we should be able to be critical and rational to trying to explore different opportunities and see the significance of those organisms and be able to collect data and being able to provide that data to future developed clinical guidelines to be able to implement NGS in laboratory settings.
Dr. Alexander, again, thank you so much for your presentation today and for your important research. And I also want to thank LabRoots and our sponsor, MicroGen-DX, for underwriting today's educational webcast. Before we go, I also want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.